Okay, let's get started then. So I'll start with one of those open-ended questions. I'm sure you heard of the central dogma of molecular biology before. If you had to describe it, you have, no? Have you not? You don't remember it? Okay, so let's discuss it. You're dying to. There you go.
My name is Asose. Central dogma, so DNA is translated to RNA or transcripted to RNA. RNA is then translated to proteins, and proteins are the way that genes are expressed.
And proteins are the way the genes are expressed. Excellent. So I'm going to come back to that. So you said DNA is transcribed to RNA and RNA is translated into protein. All correct. How do we graphically normally show that? How does the classic versions of the central dogma depict that?
Like DNA helix, is that what you're
No. What I mean is, like we say DNA, so you said DNA gets transcribed into RNA. In cartoons of the central dogma, what do you see between DNA and RNA? An arrow, that's right. So this is transcription. I will say TRX for transcription, and then RNA gets translated into protein, and that's what we call translation. Okay? Cool. And then you said that proteins are the ways that genes are expressed. Cool. Would anyone like to add anything to this cartoon of the central dogma? mRNA, what do you mean? Like you say, mRNA, what, like put it in context.
Because we can add mRNA before DNA to mRNA. So RNA is mRNA.
Oh, so we're saying that this RNA, if it's translated into protein, it should be mRNA? Yeah. Is that what you're saying? Okay. And I know you're Arshia, so I'm keeping track of participation. So what Archie is pointing out is that the RNAs that are translated into protein are actually messenger RNAs or mRNA, right? Fair enough, I'll take that. Josh?
Is there a space we could put sRNA in there?
What do you mean by sRNA?
Perhaps being transcribed in between.
But I don't know sRNA. What is sRNA?
Well, sRNA is something that is transcribed in order to change an organism's state in response to its environment quickly. And then maybe we could put like an arrow going from
DNA to sRNA. But what does sRNA stand for? I know miRNA, siRNA, snRNA, snORNA. I don't know sRNA. Small RNA. Okay, cool. So what I will do is I will say, and I will totally accept that. I will say that DNA can also be transcribed into, and I will very generically, in a way, combine all of these small RNAs that you're thinking of into small non-coding RNAs. Love it. Because, just one second. And excuse me, this is, so the central axis here is what we normally refer to as the central dogma of molecular biology. Now it's coming back. Now you just didn't know that you knew it. Yes.
Also, between RNA and DNA, it's reversible?
Between DNA and RNA. Name?
CD.
CD. All right. And it's two Ds, right?
Yeah, and HI.
HI. Thank you. Correct. So there is this process. Do you remember what that process is called? Reverse transcription. And I'm not going to draw, I don't know, I know that you didn't say either way, but I'm not going to draw a reverse arrow from small non-coding RNAs to DNA because I don't know of any examples of that. But plenty of examples of messenger RNAs that get reverse transcribed into DNA. What does that out there in the wild? Because as we will discuss hopefully soon, we can do that in a lab using reverse transcriptases on enzymes that can read DNA and write read RNA molecules and write DNA molecules out of them. So we can do that in labs. But in nature, what reverse transcribes RNAs? Certain viruses. Do you remember their name? Retrovirus, not maybe, retroviruses. And we call them retroviruses because of this ability of retro-transcribe genetic information that is written in DNA into DNA. I keep saying it wrong. RNA into DNA. Cool. So what else in nature does reverse transcription?
I was gonna do protein ribosomes.
Cool. So those are the mechanisms. Yeah, no, excellent. And we'll go back to that in a second. I just want to first wrap up this idea of reverse transcription. So retroviruses reverse transcribe RNA into DNA. What else in nature does that? We get, like, for show of hands who knew about retroviruses and how they reverse transcribed, right? So those of you who didn't know about reverse transcription probably didn't know about retroviruses either. But those of you who knew about reverse transcription and retroviruses may think that that's the only thing that does reverse transcription. And it's not. Yes?
Like, Lucia or Lucy, is telomeres, can you put it up there technically as being part reverse transcriptase? Just because
Because they do have an RNA component, you're saying, or?
it creates talons in the form of like reverse transcriptase.
But it's still using DNA as a template. Yeah. All right, I'm going to throw out a word that some of you may have heard before. I'm not sure if we're going to encounter it while building our genetic network around Alzheimer's this semester, but it is an important concept in molecular genetics of eukaryotes, and that is the concept of transposons. Have you heard of transposons before? Yeah, if you hadn't, you may have heard of this concept of jumping DNA. And the idea is that there are these DNA sequences that can move locations within genomes. And the mechanistical way in which they do it often involves transcribing themselves into RNAs first, and then reverse transcribe themselves into DNA at a different location, right? So that's another context in which reverse transcription happens naturally, right? The reason I bring all of this up is reverse transcription has become a cornerstone of many of the molecular genetic techniques that we use to measure changes in gene expression. So instead of measuring RNA molecules directly, what we do is we turn those RNA molecules into DNA molecules first through reverse transcription, and then we measure differences in the abundances of those DNA molecules that came from reverse transcription. So it's an indirect way of knowing about differences in RNA levels within a sample. Because all of that is done in labs, we tend to think that, oh, that's something that humans invented in a lab. But those of you who've seen me talk about genetics before know that I love to say that what we do in labs is always some form of hacking of what nature already invented, right? So reverse transcription is no different. So retroviruses is where we first learned about reverse transcription, but then research showed that reverse transcription happened naturally in the context of transposons as well. And just when you think in transposons, like some of you may have heard of the story of Barbara McClintock, who won a Nobel Prize for discovering transposons in corn. Okay, well, something that happened weird that happened in corn and gave someone a Nobel Prize, but who cares? We should care because our bodies are full of transposons that are constantly jumping places. So if I ask any of you what is identical throughout all of the cells in your body, what would you say? It cannot be the shape, it cannot be what they do, because a muscle cell is not the same shape, or it doesn't do the same thing as a neuron, and a neuron is not the same shape or does the same thing as an intestinal cell. So what is it that all these three cell types share that is identical? The mechanism of what? So what they do at that level? Okay, I should take it back, and then I agree with you. So these central mechanisms, they are the same, yeah. But in terms of these cellular anatomy, they're different. But in terms of what they have, what is that it's identical? Ribosomes, fair enough. And those ribosomes, these little machines that all cells have, are going to be identical. Okay, fair enough. And many of the proteins that they express, right? But in terms of DNA, right? So that's the immediate thought that comes to mind. That in essence, what makes all of the cells the same and makes all of the cells be of an individual is the genetic information that they have. They will use that genetic information to make the same ribosomes and to run the central dogma the same way, right? But ultimately, what they all share is DNA. Well, that's where I come in and say, to a certain extent, because it turns out that if we were to take cells from our bodies right now and compare their genetic sequence, or I should say maybe genomic sequence, it turns out that most cells will have identical information, of course, but you will find many examples in which that genomic sequence is not the same. Why? Because these transposons jumped places. Right? And one of the, in my humble opinion, one of the cases in which this is most extreme is in the brain, where not all neurons will share the exact same DNA, where transposons may have jumped places, which, creates this really weird notion that we tend to think of the brain as having one genetic information, but it turns out that there may be a certain degree of heterogeneity that can, in principle, contribute to heterogeneity in neuronal function, because there are these, in technical jargon, we call them mobile DNA elements. So we don't give them this old transposon name that came from corn. So now we call them mobile DNA elements. And they are constantly jumping within our cells. Okay? So let's continue with the central dogma then. And the reason I wanted to explore it, anything missing? Do you guys, particularly those of you who had encountered the dogma before, do you see anything missing in this cartoon? The central dogma is a concept that was created as a way to formalize how information flows in biological systems, right? So there was a time where people knew about DNA, knew about RNA, and knew about proteins from a biochemical perspective, but they didn't know how information moved from one type of molecule to another, right? So the central dogma came to say, well, the original information is in DNA, gets transcribed into RNA, and RNA gets translated into proteins that are the ones carrying those biological functions. I think you said that proteins are the expression of genes, right? So I'm going to say this. I'm going to add this arbitrarily, and I'm going to say that this leads to phenotypes. Phenotypes being the physical manifestation of genetic information that is stored originally in DNA. Right? But then came all of these exceptions that started popping up all over the place. Like, well, hold on a second, because sometimes genetic information can flow back from RNA to DNA. Hey, hold on a second, not all RNAs are getting translated into proteins. Right? So, and one of the lines that I would like to draw now that is not very common in most classical depictions of the central dogma is this one here. And again, I asked Josh, right? Yeah. So that was a very important contribution, because this arrow from small non-coding RNAs to phenotypes is something that is relatively new. The central dogma was put together in the 60s. The contribution of functional small RNAs to phenotypes is something that we've known for perhaps the last 40 years or so, right? Much more recent.
4 years?
40. Yeah. So for a while, even though everyone knew, Arshia pointed out, well, the RNAs that are translated are the mRNAs, and we've known forever, from about the same time that we learned about mRNAs, that there were these two other kinds of RNAs, the two other major classes, the transfer RNAs and the ribosomal RNAs. But those were like, well, those are accessory RNAs. They don't really do anything by themselves. They're just accessories to translating mRNAs into proteins and therefore exerting function. It was many decades later that we learned about small RNA molecules that do not translate into protein and yet have biological effects that amount to phenotypes. What I mean by that is there is small RNA molecules that, when present in a biological system, sometimes expressed by the biological system itself, sometimes coming from outside, will affect the way that biological system functions, and therefore they will be bringing about a phenotype. Okay. Again, I don't know if we'll encounter any examples of these or not, but as we will see probably on Wednesday, there is a big family of small non-coding RNAs called microRNAs, and microRNAs have the ability to exert phenotypes in biological systems. So will we encounter any microRNAs in our Alzheimer's / dopamine / sleep network? Maybe. We'll see. We'll see. But these things are huge. Anything else, anyone? There's one tiny little arrow missing from the classical cartoon of the central dogma. Can anyone think what that is?
DNA replication.
Exactly. And I didn't get your name last time.
Annalisa.
Annalise. Correct. So there is that bent arrow of DNA going into itself, and the core message there is that all the information that biological systems need to make new DNA, well, not all of it, let me put it this way. You can make DNA out of RNA when there is a retrovirus or when there is a transposon going around, but in the absence of those, all of the information that you need to make new DNA is in DNA itself. Okay? Cool. So that should do it. That should do it. Any questions, anyone? Yeah.
You pointed the arrow to the small non-coding RNAs to the phenotype.
Mhm.
I thought that those were like regulatory.
Correct.
Okay, so, but they don't actually produce like
The phenotype themselves. Excellent point. Thank you.
I just want to make sure, because that was.
Absolutely. No, let me clarify. That's an excellent point, and I'll clarify. So and your name? Alejandra. What happened? There you go. You say your name and the lights went on. That's weird. Alejandra's point is that these small non-coding RNAs, they're not exerting phenotypes the way that we typically associate with proteins, like catalyzing chemical reactions, moving things around. That instead, these small non-coding RNAs serve regulatory functions in, let's say, the central axis of the central dogma or the middle axis of the central dogma. So it is somewhat indirectly that they affect phenotypes. So, for instance, small non-coding RNAs may regulate the abundance of mRNAs, or they may regulate processes like translation. Most importantly, they may regulate, I'm going to make this arrow a little thicker, they may regulate the process of transcription itself. So one way to look at this is they're not carrying out any biochemical reactions or processes by themselves. They're just telling others when they should do or not their thing. And I agree with that, Alejandra, completely. But I would argue that is often what happens with proteins anyway. So when we say that a given protein is associated with a phenotype, that protein leads to that phenotype in the context of many other proteins and other molecules that are around it. Does that make sense? So when we say, for instance, that a given protein is associated with the ability to digest an amino acid, right? And that the lack of that protein might make the individual sick whenever their diet includes that amino acid, for instance. So the protein, or the lack thereof, is not contributing directly to the phenotype. It is embedded in a system of biomolecules that now make the absence of that protein have an effect in the physical well-being of the organism. So even for proteins, the link to phenotypes is really relational, in that proteins exert their phenotypes in relation to other proteins within cells. So in that sense, small non-coding RNAs are not that different, and we say that they exert phenotypes in the sense that through the regulation of many other things that are present in the cell, they have an impact on the physical manifestation of the gene information in that system. Does that make a little better sense now?
Yeah.
The distinction here would be, for instance, transfer RNAs and ribosome RNAs, which are, in some like weird philosophical sense, completely agnostic. They will make one protein or another. They don't care. Does that make sense? Yeah. Whereas these small non-coding RNAs have very specific regulatory functions, I totally agree with you, that bring about a specific phenotype. We took it from 0 to 90 in no time, but did that make sense for everybody? Yeah? Cool. All right. You guys ready for there's no way we're going to make it through the whole lecture at this rate, but this is cool. This is exciting conversations. Guys ready for the next open-ended question? And now I asked the same question. So how would you guys describe the structure of a typical eukaryotic gene, right? I'm going to ask those of you who already participated, maybe hold back if you can, so that everybody can speak up. So how do we, if you have to like draw a eukaryotic gene, how would you draw it? 5 prime to 3 prime, okay. And we begin where? Yeah, but what component of the gene? The what? The promoter. Excellent. So you're saying, and your name? Jacob. So you're saying we started a promoter that is somewhere in the 5 prime end of a gene, right? And what comes after the promoter? What's in the gene? If anyone else wants to chime in.
I don't remember exactly what comes after the promoter,
Exons and introns and exons. Okay, do you remember which one comes first? Yeah. This is a rule that if you guys want to remember, all genes or all transcripts start with an exon. There's no way that you can start with an intron. There's reasons for that, but it all starts with an exon. So let's say some exons and some introns. And for simplicity, we'll keep it at 2 exons, 1 intron in between.
I would probably start with drawing the double strands of the DNA and then the untranslated region right before.
The double strands,
Or because this is of the DNA.
yes.
And then, so yeah, I would put this strand and that beginning is put the untranslated. Oh. Well, that's on one, isn't it? The promoter's on one of the strands. You put the strand.
I appreciate that comment. So you said the promoter is in only one of the strands. When we're thinking DNA, with the only exception of DNA replication where the strands are separated, right, from one another, when we're thinking DNA, it's almost impossible to separate the strands from one another. What I mean by that is that the information in one strand is reflected or complemented by the information in the other, right, by the natural biochemistry of DNA. And as far as we know, what the molecules, and I'll be a little more specific, what most of the proteins that interact with DNA, quote unquote, see is actually the combination of both strands. They don't see letters, they see base pairs. Does that make sense? Biochemically speaking, when a protein interacts with DNA, again, the only exception would be DNA polymerase when the strands are separated during DNA replication. But when proteins are interacting with native DNA in its double helical structure, what they see is the double helix. And what they will do is read base pairs. For simplicity, when we are describing a promoter, we may describe the sequence of letters in one of the strands. Because it's a little annoying to, instead of saying this promoter has a TAA, TTA, it's a little annoying to say a TATA, AT, TATA, ATAT, right? So we cut ourselves some slack by reading the sequence of nucleotides in only one strand. But that is not to say that the protein interacting with that region in the cell is reading only one strand of DNA. It's reading both. It's reading the region, okay? All right, so let's take ourselves back. So we have the DNA double helix with a promoter at the 5' end of the gene. Then there is a sequence of exons and introns and exons. Where does this gene end? Louder, I saw you.
Terminations or stop point at the very end.
So you said two keywords, termination and stop. Do you want to pick one?
I'll say termination.
Termination, excellent. So the idea is that somewhere here there is a termination signal. Yeah? Everybody agree with that? Does someone disagree with that? All right. Well, you just said your name? Lucy. Lucy. What you just said is true in bacteria, in prokaryotes. Some eukaryotic genes do have termination signals. Most protein coding, mRNA producing genes do not. As we will see later today, if we have time, eukaryotes evolved an alternative to the termination signal. So strictly speaking, termination signals do not belong in your typical eukaryotic gene. Instead, what happens is eukaryotic RNA polymerases, the enzymes that are going to be reading an RNA molecule off of this gene, will read past the end of that gene and make a much longer than needed RNA molecule. And then there is a process that comes afterwards, after transcription has completed, that will cut that longer RNA shorter and process that RNA in such a manner that it makes it ready for translation eventually, okay? But we'll discuss that in a minute. So what else it might be missing in this structure, the typical structure of a gene? Is that in DNA though, or is that something that happens to the RNA? Yeah. Regulators, okay. What do you mean by regulators?
In order for a gene to be expressed, we need certain regulators,
Uh-huh.
right?
Excellent. Yeah. I'm messing with you. No, absolutely. Yeah. So, but wasn't that what the promoter is for? I already told you that. You're absolutely right. So, now you can just go with it.
Aren't there like regulators that bind the DNA polymerase to a promoter?
And
Recognize it?
exactly. So now you said a key word. So you said regulators that bind the RNA polymerase to the promoter, right? So that's how we, in a way, define promoters. Promoters are the sites where RNA polymerase binds and starts its job. right? So here's where the RNA polymerase will initiate its business. But then these regulators, I'm sorry, I asked you your name before? Oh, no, I didn't ask you your name before. And yours? Melanie. Welcome, Melanie. Now I put a face to the name. So the regulator sequences in DNA that are part of a gene, so I'm going to include them as part of this gene, are where some regulator proteins bind, whatever that was, and I'm going to refer to those as activators and repressors, and they will either facilitate or inhibit the binding of RNA polymerase to the promoter. Right? So the promoter is where RNA polymerase starts its business. These regulator sequences that are part of genes are collections of DNA sequences that will facilitate the binding or mediate the binding of activators and repressors, which in turn facilitate or inhibit the binding of RNA polymerase to that promoter. Okay? Cool. How about the structure of a typical eukaryotic mRNA? So now DNA is behind us, so somewhere up there. I drew it as a line, but we can imagine the double helix in there. And now we know that there's been transcription. So if you were to draw a typical eukaryotic mRNA, messenger RNA, what would you do?
Just include the extrons.
Just include the exons. What happened to the introns?
They'll cut out.
Cut out. Okay, so I'm going to say exon, and then the intron is gone. Something like that? Cool. The cap and the tail. I know, Gabi, you said it first. Name? Andre. The cap and the tail. So the cap, we'll discuss it no, we won't. So the cap is a chemical modification of the very 5' end of mRNAs that all mRNAs undergo, and is important for cells because it tells them that the mRNA that they're kind of dealing with is intact, that it hasn't lost any of its sequence. So the presence of a 5' cap tells the cell this is a healthy mRNA on one end. On the other end, there is another signal that cells use. And Andrea, do you mind elaborating? You said the tail. What do you mean by that?
There's a 3'
polyadenylated tail or poly-A tail. Yeah, so you guys must have seen this many times. At the very end of eukaryotic mRNAs, there is this enzyme that will cut that very long primary transcript short, and in the process, it will add, without asking, totally arbitrarily, it will add tens, if not hundreds, of A's, adenines, to that 3' tail. Right? And these two are used by cells primarily as ways to know that that RNA is legit, is intact, came from the cell itself, that the cell itself produced it, right? So you shouldn't go destroying it. And as we will see, they're also used as signals by the ribosome that the RNA is whole. So it's not only that it's not an RNA virus invading the cell, but also that if we are going to translate this thing, we have some sort of reassurance that the message is intact and it didn't break along the way. Huh? Cool. So yeah, the typical structure of your eukaryotic mRNA would be a 5' cap, a poly-A tail at the 3' end, the exons without the introns between them. That's spotless from a sort of a biochemical structure point of view. Information-wise, so where does the code for the protein begin here? Say it again. The code starts at the 5'. Any other ideas, anyone? The first exon, so as I said before, the all genes start with the first exon. So the beginning of the first exon would be, I suppose, here. So are you guys saying the same thing, that the AUG, the start codon, is at the beginning of a molecule? Anyone a different idea? No? I don't know who to go. Oh, okay, so, okay. But you promised that if you thought the same thing, you're going to share credit? Okay, go for it.
Doesn't it, like, does it matter which way it starts?
Yeah.
Does that make sense?
Is that what you had in mind?
I didn't hear what you said.
That it doesn't matter which way it starts. What do you mean by that? I'm curious.
I just remember there being like it doesn't have to start from the five.
Oh, okay. So you're saying it doesn't need to be at the very beginning.
Yeah,
It can be anywhere.
yeah.
Ah, all right. Arshia?
I think the direction matters, but it could be a different exon.
Okay, so two things important there. The direction matters, so the star will simply say where it starts, but then the direction of the cells read the message matters. I totally agree. And it could be on a different exon, and that's absolutely true. So here in this cartoon mRNA that we just produced, there were two exons, remember? There's nothing against the AUG, the start of the protein coding sequence, being in the second or third exon. That can happen. Most eukaryotic genes that have many exons have many exons. We're talking like sometimes tens of them. And it is common for the AUG to not show up until exons 5, 6 or so. Yeah? The message here is that the beginning of the mRNA is not the beginning of the protein code. The beginning of the protein code is established by that start code that we refer to as AUG. For a simple reason, that's the start code universally, right? And where does this message for making a protein end? At the stop codon. And where is the stop codon? At the poly A tail?
It has to be like a UAA,
One of those. Where are those? At the very end? Middle to end. I like that. Before that poly A tail. That poly A tail means nothing really, right? Code-wise. I mean, it could mean the same amino acid over and over, but that's not the point. The point is that the stop codon, whichever it is, it's 3 of them, will be somewhere before. Cool. So this gives eukaryotic mRNAs different regions that we name differently, right? So the sequence that is between the AUG and the stop, we refer to for short as CDS, for coding sequence. What do we call anything that is upstream of the AUG? The UTR, untranslated regions. And there is a 5' UTR and a 3' UTR. Okay? It is the most common human mistake because humans are not instinctively, they haven't evolved to understand the structure of eukaryotic mRNAs. The most common mistake is to confuse the AUG with the beginning of the mRNA and the stop with the end of the mRNA. And the same goes for at the gene level. So the beginning of the transcribed region of a gene at the DNA level is the transcription start site. But that is not the AUG. The ATG that codes for the AUG in the mRNA will be somewhere downstream. And it could be, as Arshia was pointing out, in any of the, it doesn't have to be the first exon, it can be any of the posterior exons, okay? Or later exons, that's what I should say. Okay? Cool? So, not a whole lot going on with 5' UTRs, other than perhaps at the level of translational regulations. So there is information in 5' UTRs that will say to a cell, you know what, we have this mRNA, but let's hold on translating it yet, because for whatever signals I'm getting, we don't need to translate this yet. Let's keep it. Perhaps even send it to a specific area in the cell, but let's not translate it just yet. A lot more fun going on here at the 3' UTR, as we will see on Wednesday. That's where the regulation by some of those small nuclear RNAs, like Josh and Alejandra were pointing out, happens, and we'll discuss the molecular mechanisms of that. Questions, anyone? Comments? Nothing along the lines of, I once heard that. No? Okay, cool. All right, so this slide you have in your decks is just summarizing everything we just said. So there is a gene in DNA that has a promoter. That particular cartoon is not showing the regulatory regions, by the way. There's the exons. All genes begin to be transcribed at exon 1, and there is the TSS, transcription start site. There is all the exons and introns. There is no no 3' termination signal. There is transcription, and then there's a bunch of things that are going to happen. The introns are going to be spliced out. There's going to be 5' capping and polydylation at the 3' end that is dictated by a cleavage site or a poly-A signal. And then after all the introns have been taken out, we end up only with exons. But exons do not equal protein coding sequence, because exons simply mean I'm going to be there in the mature mRNA molecule. That's all it means. Being an exon doesn't guarantee that you carry protein coding sequence. It just says that you'll be there in the final product. The sequence that codes for the protein, or the sequence of amino acids, is going to be the CDS, starts with the AUG, ends with any of the 3 stop codons. Anything upstream of that is the 5' UTR. Anything downstream of it is the 3' UTR. And once the ribosomes go and translate through this mRNA, you'll get a protein. Okay? So one thing that I should say is that we talked about the typical structure of a eukaryotic mRNA. I can emphasize that enough. Everything that we just discussed is for mRNAs. Non-coding RNAs, including ribosomal and transfer, they don't have 5' UTRs and 3' UTRs because they don't have a translated region. So how could they have an untranslated? The whole thing is untranslated, right? So everything that we just discussed here applies to mRNAs. Okay, so let's move along. Can someone tell me at what levels of the central dogma cells regulate the expression of their genes? And once again, if I haven't heard from you yet and you want to chime in. So how can a cell decide if and how much and when it will express a gene or not? So how can a cell regulate the expression of its genes? Histone proteins. So what do histone proteins do?
They wrap DNA.
And how does that regulate the expression of a gene?
Regions between histones are usually expressed.
Uh-huh.
So it kind of regulates like what section of DNA.
And when you say that those regions in between histones are expressed, how are they? Because you're talking DNA so far, right? So how does DNA express a gene? But what needs to happen to DNA for it to express a gene that is present in it? What needs to happen with that gene? Transcription, right? So what you're referring to is making that DNA accessible such that a cell decides if, instead of saying, a cell decides when and where and how much it wants to express a gene, we simply can say when, how much, and where it transcribes a gene, right? So there is the transcriptional level. So there is transcriptional regulation. What else? It has a cell has transcribed already a gene. What else can it do to it? Translational, right? So a cell might decide, as we were discussing just now, whether to translate an mRNA that it already has or not. That's a point of decision that cells regulate.
Post-translation?
Post-translational, what does that mean?
Like with proteins, they like components their function and stuff.
We'll discuss that with one slide on Wednesday, but absolutely correct. So we tend to think that, or people tend to think when learning about translation and whatnot at first, that once an mRNA has been translated into a protein, we're done. That protein is off the, what's the expression, of the gate to do its job, right? Not quite. Many proteins require a series of post-translational modifications, meaning chemical modifications that happen after they've been translated, for them to acquire that biological function, for them to be able to exert that phenotypic effect in the system.
So wouldn't there be like a post-trans, like, how do you say that word?
Transcripts. What,
Post-transcript?
did the gap give it away?
Yeah.
Absolutely, yeah, of course there is. Post-transcriptional.
I was wondering why was it
Post-transcriptional. What that means is these are mechanisms that cells can use after they've transcribed a gene, but before they translated it, to move the process along or not. So post-transcriptional regulation of gene expression may have to do with maintaining an mRNA around or actively degrading it. It may have to do with making different flavors of the same mRNA or the mRNA that comes from the same gene.
microRNA and silencing RNA?
Correct. Those would fall in here within this category. These are mechanisms, regulatory mechanisms that affect the expression of a gene after the gene has been transcribed, but before it has been translated. Yes. Yes. And then, sorry, I didn't catch your name before. Helen. Helen, you mentioned histones, and this is actually what we'll discuss with whatever is left. There is a step that, in a way, this is debated, so I'll preface with that. But there are some people who, me among them, who like to think about pre-transcriptional regulation. And what do I mean by that, or what do we mean by that? That when we think of transcriptional regulation, we're thinking about that last-minute decision to transcribe or not. Simple. The cell decides to either bind an RNA polymerase to that promoter and let transcription begin or not. But often that decision is preceded by other regulatory mechanisms that may make those genes available to the cell or completely unavailable to it. And those mechanisms can be sort of classified as pre-transcriptional.
Do energy levels fall under pre-transcriptional, as in like when there's not enough energy within the cell, it chooses not to?
Wonderful question. And I will answer by saying that energy levels, so Gabi is basically saying sometimes energy levels can make a cell decide whether to express certain genes or not, right? I will place energy levels into a category of regulatory inputs. So it could be energy levels. It could be certain signals from the environment. It could be certain metabolites produced by the function of the cell that is telling the cell you're doing a little too much of this. It could be certain nutrients. It could be specific hormones coming from somewhere else in the organism or a different organism. Right? All of those things I will bag together as regulatory inputs. And those regulatory inputs tend to impact the expression of genes at all levels of the hierarchy. There will be inputs that have to do with energy levels that happen pre-transcriptionally. Energy levels can also affect how certain proteins are phosphorylated, for instance. So they will typically hit any of these levels. Cool. Yeah. All right. So this is a cartoon basically drawing what we just said. We start with, there may be areas of DNA containing genes that a cell might like to express at some point, but exist in what we call a very compact conformation. So there has to be an alteration of that structure to relax those regions where those genes are. So that's part of that pre-transcriptional regulation. Then cells will make decisions about whether to transcribe genes or not, giving rise to pre-mRNA molecules that are processed, and that's another point of regulation because mRNA processing is one of the entry points for regulation, for post-transcriptional regulation. Then once the processed mRNA is all said and done, there is regulation via RNA stability or the regulation of translation initiation, this business of a cell may have an mRNA that could be translated right now, but it will decide to hold on until a later time, and then post-translational modifications that affect ultimately the function of those proteins, right? So what I'm doing here on this slide and all the subsequent ones is keep that map of the levels of gene expression regulation to the left, so that we sort of know where we are in the process. So let's talk about that pre-transcriptional regulation. Turns out that DNA, in general, is, as Helen was pointing out, wrapped around these very special proteins called histones, right? Histones don't necessarily function as single units. They function as octamers, so it's groups of 8 histone proteins working together, and they have this peculiar composition of 2 H2 molecules, 1 H3, and it's 2 H2As, 2 H2Bs, 1 H3, or 2 H3s and 2 H4s, so total 8. And these octamers serve as some sort of like core around which DNA wraps itself, right? Now, what happens is each of those histone octamers with DNA wrapped around them. Helen, do you remember what those are called by any chance? What these histone octamers with DNA around them are called? Chromatins? Chromatin is the whole thing. Chromatin is the mixture of DNA and proteins. What do we call these things? You guys remember? This unit of a histone octamer with DNA wrapped around it starts with an N. Yeah? Nucleosome. There you go, nucleosome. Yes. So this is a nucleosome. It simply means a body in the nucleus, right? So a nucleosome. And these nucleosomes can be in a relaxed conformation. So they're, the term that we normally use is strings on a bead. Beads on a string, right? So beads on a string, like almost like a necklace, but it's loose, right? So you have nucleosome and then some spacer DNA and then nucleosome and spacer DNA. But every once in a while, cells or nuclei decide that they need to compact these nucleosomes much more tightly. The thinking in part is, if I'm not going to be using this DNA for a while, or the information contained in this region of DNA for a while, I need the space, right? And this thing that is so loose and relaxed occupies a lot more space. That's something that is much more tightly compacted, right? It's almost like spooling a string really tightly around some sort of axis just to make space. So those are the two conformations of chromatin. chromatin is that collection of nucleosomes strung to each other by DNA. So chromatin can exist in these two conformations. Euchromatin is the one that is relaxed, where things are a little more accessible. The information contained within that region is a little more accessible to the rest of the cell. And then heterochromatin is the highly tightly compacted form of chromatin that makes everything very inside and inaccessible. So in heterochromatin, DNA is inaccessible and genes are unavailable. In euchromatin, DNA is accessible and genes are available, but they're not transcribed yet. So part of this business, it's only part of it, but an important part of this pre-transcriptional regulation of gene expression is for cells to decide which regions are going to be euchromatic or heterochromatic. So regions that do not contain any meaningful genetic information for a cell at any given time may exist in heterochromatic regions. The rest will be most likely as euchromatin. But like I said, euchromatin makes genes available but not necessarily expressed. They're not yet transcribed. For a gene to be transcribed, RNA polymerase cannot have these nucleosomes in the middle, right? So when the cell decides that it wants to transcribe a gene, it needs to first fully expose that promoter where RNA polymerase will start its job, and then devoid the rest of the gene of any of these nucleosomes, because RNA polymerase can go around bumping into nucleosomes, into histone octamers all along, right? So what happens is that when a cell made a decision that a certain region needs to be transcribed, it will use something that we call a chromatin remodeling complex. It's a bunch of enzymes that consume a lot of ATP, a lot of energy, and those chromatin remodeling complexes will essentially disassemble nucleosomes and will leave behind what we refer to as naked DNA. So now we can start thinking of the double helix. Yeah? So when a certain region has become a nucleosome free, that's when the RNA polymerase can bind to the promoter and start transcription. Separate the two strands and read one of those strands of DNA, the template strand, and make an RNA molecule that is complementary to it. Right? We're not going to review the molecular process of transcription in this class. So if you need a refresher on that, gazillion videos out there explaining how that works. Okay? So euchromatin makes genes available but not transcribed. For genes to be transcribed, they need to be depleted of nucleosomes, and that's the job of the chromatin remodeling complex. Now that creates a bit of a paradox. This was from one textbook that I used to use for teaching the 300 level genetics. But the text that you see here, you can find in pretty much any textbook. And it goes something like this. The chromatin remodeling complexes are targeted to specific DNA sequences by transcriptional activators or repressors that are bound to the promoter of specific genes. And that creates a paradox, because what that statement is saying is, for the chromatin remodeling complex to know where to start taking out nucleosomes, it first needs activators and repressors binding to DNA. But didn't we need to take the nucleosomes out for these things to bind the promoter? What's first, the chicken or the egg, right? There seems to be a little bit of a contradiction there. So if the CRC, the chromatin remodeling complex, needs to expose, TFBS there stands for transcription factor binding sites. Before any activators or repressors can read the promoters in DNA, how can these activators and repressors direct the CRC to specific genes? So there seems to be a paradox there. The solution is this, that it turns out that for RNA polymerase to transcribe DNA, it needs full access to naked DNA. No question about that. It needs to separate the strands. The RNA polymerase is a huge machine that needs to move through the space without any nucleosomes. But for transcriptional regulators, like these activators and repressors that I mentioned here as the first egg, if you will, they don't need access to naked DNA. What they need access, at least, is to the sequence in the double helix, right? Such that they can access the DNA double helix relatively unimpeded from the outside, if that makes sense. And we go back to this idea that DNA reading proteins don't read one strand. They read really the double helix. They read base pairs. Now, that is accomplished by simply loosening a little bit how DNA is wrapped around these histone octamers. So I mentioned before that chromatin can exist in these very two conformations, these two distinct conformations, heterochromatin and euchromatin. What I'm about to say can be a little confusing sometimes, so I need you guys to listen up. One thing is heterochromatin versus euchromatin. That means that string of beads tightly compacted in heterochromatin or kind of loose in euchromatin. But then you can look within each of these nucleosomes and take a look at how tightly wrapped DNA is around those histone octamers. In its natural conformation, DNA and the histone octamer are very tightly wrapped around each other, or DNA wraps very tightly around the histone octamer. And the reason for that is histone proteins have a lot of positive charges in them. And what do positive charges love above all things? Negative charges. Where can we find negative charges in DNA? Does DNA have any negative charges? Where? The phosphate backbone, right? If you guys remember, your DNA is two strands of nucleotides strung to each other by phosphodiester bonds. We call that the phosphate backbone. Nothing but a collection of negative charges there. No sequence to be found there. It's just phosphate, sugar, phosphate, sugar, phosphate, sugar, right? The fun part, the sequence part, happens on the basis side of things. So the positively charged histones interact very tightly as ions with the negatively charged DNA. And that makes for this very close approximation of DNA to histone, the histone core. If cells want to make certain regions of that DNA a little more accessible, what they need to do is make that DNA let go a little bit of the histones. And how do they do that? By neutralizing the positive charges in histone. There is a number of chemical reactions, mostly, or if not, primarily acetylations and methylations, that will take the positively charged residues or amino acids in these histone proteins and modify them chemically such that now they became neutral. So when positive faces negative, they bind to each other very tightly. They love each other, right? But when you have a neutral and a negative, they don't care about each other anymore. And DNA will still be wrapped around histone, but much more relaxed. And that is what makes it accessible. That's what makes DNA accessible to some of these DNA binding proteins that all they need to do is access those regions of DNA where the TFBSs are, the sequences of nucleotides that they can recognize and bind to. So that is how these transcriptional activators and repressors can bring the chromatin remodeling complex that can then take out the nucleosomes and allow for the core transcription factors and polymerase II to begin its job. Right? So I'm trying this cartoon out for the first time with you guys. Let me know if it works. So you can think of euchromatin as the DNA is accessible and genes are just available, and that's sort of the ready when you're about to start a race, right? Then comes the loosening of the nucleosomes by modifying the histones, by acetylating them or methylating them such that the attraction between the histone and DNA becomes a little bit more relaxed. So now the gene is ready to be transcribed. When the right activators, the right combination of activators and repressors assemble at a given promoter, let's say that this is the gene that we'd like to transcribe and the promoter is somewhere here. That promoter now, in these more relaxed nucleosomes, may be more accessible, and I should say the promoter and also the regulatory regions, right? Let's do yellow so that we can see. So let's say that these regulatory regions and promoters are buried somewhere in those nucleosomes. By methylating and acetylating the histones in that particular nucleosome, we can make those regulatory sequences and the promoter a bit more accessible. And now these transcriptional activators, I won't say repressors anymore because that's counterintuitive. So let's say that the transcriptional activators will bring the CRC, the chromatin remodel complex, which will take out all of the nucleosomes and now expose fully naked DNA for RNA polymerase to come in and go with the transcription. Okay? So there is making the gene ready, poising the gene for imminent transcription, the get set, and then finally when the decision is made, the go. Yeah? Does that work? Sort of a metaphor? Cool. So, we'll spend the last 3 or 4 minutes going over this slide, and then I'll figure out how we rearrange the rest of the slides. We can probably move them. If we need to spill over next week, we will. I want you guys to learn. So if I ask you, and if you didn't see me write your name down, this is your time to shine. So if I ask you, are histone modifications, these acetylations and methylations that we talked about, are they reversible or irreversible? Let's see if someone who didn't participate today. Did everybody get a chance to speak today or ask a question? Yes, Ale. You say reversible. So meaning that what we do to a histone, we can undo. Right? What we do to a histone, we can undo. And that's absolutely true. That's correct. These histone modifications, acetylations, and methylations are reversible. Just like there are acetylases that will stick an acetyl group in these histones, there are deacetylases that can take them out. Same for methylases and demethylases. How about unstable or stable? Are these modifications stable or unstable? Unstable? Why would you say unstable? I see where you're coming from, but I want you to elaborate the logic behind it. Exactly, you said it. You just said it. There. If they're reversible, then it means that they're unstable. That once they happen, they will unhappen, right? Yeah. Yes, but and here's where the a little bit of the counterintuition comes in. Even though they're reversible, they're very stable. What I mean by that is that yes, any acetylation can be deacetylated. But that's another decision, active decision made by the cell. What I mean by that is when a cell decides that it wants to express a gene, it will go and acetylate the nucleosomes around the regulatory region of that gene, right? But it's not like the deacetylases will sneak behind and start deacetylating everything the acetylases did. Exactly. So these modifications are reversible, meaning a cell can change its mind and change the status of a given nucleosome, but that is a second active decision process. Until that decision is made, these modifications are very stable. And they're very stable for the very simple reason that they are covalent chemical modifications of histones. Things that are things are unstable in biology when there is no covalent bonds, when interactions are kind of wishy-washy, like hydrogen bonding or ionic interactions, right? But the second you establish a covalent bond, you created a chemical link between those things. That acyl group won't go anywhere, right? Until the cell decides, you know what, I changed my mind, I want to take this acyl group out. So then the modification is deacetylation, and that modification will again be reversible because it can go back to being acetylated anytime later, but it will also be stable. Once deacetylated, that histone will remain deacetylated until the cell decides so. And here's the kicker. Are these modifications inherited or not inherited? I see some heads shaking like that. Not inherited? It depends, I see. Depends on what?
So, because from what I know in another class, had to do with epigenetic markers, if it adds a methyl group and it maintains it for a couple generations.
Yeah, you said.
It just depends on what it adds on to the DNA.
You said the E word, epigenetics. Yeah, what does epigenetics mean?
The DNA gets passed on, and the original, let's say like the mother or hermaphrodite,
Uh-huh.
might have something, it modifies the DNA, it passes it on, usually through a system where the mother experiences it, and it uses a different RNA system to go in.
I agree 100% with everything you said, but let's take it back so that we're all on the same page. So when I say genetic information, you guys should, by now, start thinking A, G, T's, and C's, right? So the sequence of nucleotides in DNA that get passed down generation after generation. Hopefully, faithfully, no mutations, the best we can, right? So that's genetic information. It turns out that there is epigenetic information. Epi means on top, right? So epigenetic information is all of these modifications that are done to key nucleosomes, and yes, they can be inherited. And that's how organisms can pass down to their progeny, and then cells can pass down to their progeny as well. When a cell divides, it can tell its daughter cells what type of experiences it lived, and therefore what the daughter cells should be prepared for. So if I'm an intestinal cell that is making an enzyme for digesting lactose, right? And I'm about to divide, excuse me, and make two of myself, I would like to tell my daughter cells, you should make lactose. So how do I do that? My daughter cells will have the gene for lactase, the enzyme that breaks down lactose, but I should be able to tell it to use it. We do that through epigenetic modifications. These are very stable modifications of histones that can be passed down over generations. So yes, they can be inherited, and that gives rise to the concept of epigenetics.
From the daughter
Mother-daughter cell, so it goes from the mother cell or progenitor cell to its descendants.
Yeah.
We use mother cell, daughter cell loosely, but the more technical term is progenitor to descendant.
Wouldn't it be like 50-50 because it's reversible, it's somewhat stable, kind of stable? Is it like a...
So I'll address the question. So everybody, we're a couple minutes past. I want to be respectful of your time. So I'm going to address this question if you want to stick around. But if you need to go, I'll totally understand. I'll see you on Wednesday. Okay? No, because once once a modification has been in place, by the way, if you need to go, I'm going to record this and you can see it in the recorder later. So don't feel pressure to stay right? Cool. If a modification has been made and is stable, when I say stable, I really mean it. It is stable, right? So the progenitor cell had that modification, and now when it creates two daughters, it needs a way to duplicate that modification. It needs to make sure that both daughters inherit the sequence of the gene and also these modifications that were made to the histones where the gene is. The mechanisms for that, they're not super intuitive, but they're known, right? The idea is, the thinking is, when a cell is replicating DNA, it will separate the strands of DNA. To do that, it needs to take the histones away because it cannot work with histones around. But those histones remain nearby, okay? So what happens is when imagine there is this gene in DNA that is wrapped around this histone, and this histone was heavily acylated. Okay? So now the replication machinery comes in and says, okay, I need to replicate through here. So if you don't mind, I'm going to put this histone to the side, make 2 copies of DNA. I will use the old histone, of course, the old histone octamer to make a new nucleosome, and I will use it as a template to see what modifications I should make to this one. Did that make sense?
Yeah.
I super oversimplified it, but the idea is that when cells are replicating DNA, they have a way to replicate the epigenetic code. Right? Each of these nucleosomes will have a collection of acylations and methylations that mean things to cells, that communicate a state of the progenitor cell, but they're reproducible through cell divisions. To the point that they're reproducible through gametogenesis. So when sexually reproducing organisms are making gametes, they can print some of these epigenetic signals in their gametes. So now we can take it to the next generation. Yeah?